Welcome to my YouTube channel, Everything Medical. Today's video is all about metabolism. It's all about the production of glucose from non-carbohydrate precursors. Before we continue with the class, with the video, if you are coming across my channel at the first time, don't hesitate to subscribe. Yes, endeavor to subscribe and also comment, share our video. After watching it, you comment, you still share. It will help those who are preparing for medical board exam. So let's proceed. Today's video is all about metabolism, that is the production or the synthesis of glucose molecules from non-carbohydrate precursors. This pathway is essential for the fact that our body do be in a state of starvation or fasting, or in a case of diabetic mellitus per se. One thing I want you to understand in this pathway before I proceed is that gluconeogenesis, which is the pathway we are treating, occurs during starvation or fasting, or in a case of diabetic mellitus. It occurs during starvation or fasting, or in a case of diabetic mellitus, right? It does not occur during well-fed state. It does not occur during well-fed state because there is no availability of glucose. That is why it is being activated to produce glucose for continuous tissue respiration or tissue metabolism, right? Before we talk other key facts about this pathway, let's give it an introduction. Right? What is gluconeogenesis? Before I proceed, in here is the cell cytoplasm. This is the cell membrane, right? The cell membrane or the plasma membrane that is protecting the cell cytoplasm or the cytosol. Why this is the cell mitochondria, right? Suspended in the cell cytoplasm. You have to understand that metabolic pathways or metabolism usually take place in two regions of the cell. It's either it's occurring on the cell cytosol, or it is occurring in the cell mitochondria, or it is occurring in both regions, right? So as we proceed, we'll get to know which cellular compartment is this pathway taking place. Now, back to the introduction. What is gluconeogenesis? Gluconeogenesis is the synthesis or the production of glucose molecules from non-carbohydrate precursor. It is the production or synthesis of glucose molecules from non-carbohydrate precursor. I said previously that this pathway occurs during starvation or fasting, or in the case of diabetic mellitus, because insulin is not active. Insulin is not active during starvation or fasting, or in the case of diabetic mellitus, right? So that is why high glucagon, low insulin ratio is potentially gluconogenic. High glucagon, low insulin ratio is potentially gluconogenic, right? Why high insulin, low glucagon ratio is potentially glycolytic? That is to say that this pathway is being favored by glucagon by activating its key enzymes. Why insulin does not favor it? Rather, insulin is a counter hormone in this pathway, because this pathway has a hormonal regulation, right? Insulin is glucagon is a current hormone, thereby activating their key enzymes, right? But then, insulin is a counter hormone, thereby deactivating its key enzyme in order to inhibit the pathway. That is why you have to understand the countercurrent hormones between the countercurrent reaction between insulin and glucagon, right? So high glucagon, low insulin ratio is potentially gluconogenic, why high insulin, low glucagon ratio is potentially glycolytic, that is for glycolysis, right? Because gluconeogenesis is the opposite of glycolysis, but not a reversal of glycolysis. That is what you should understand. That gluconeogenesis, this pathway, is an opposite of glycolysis, but not a reversal of glycolysis, right? Because the key enzymes of gluconeogenesis circumvent the key enzymes of what? Of glycolysis. That is why it is an opposite of gluconeogenesis, of glycolysis, but not a reversal of glycolysis. So we have given a concept about this pathway, that it involves the synthesis of glucose from non-carbohydrate what? Precursors, right? So the next outline we shall be looking on to is the cellular compartment. Cellular compartment, right? When we talk about the cellular compartment of a metabolic pathway, we refer to the part of the cell it occurs. Gluconeogenesis is partly cytosolic and partly mitochondrial based. That is to say that it occurs in the cell cytosol and also occurs in the cell mitochondria. Similarly, its enzymes are partly cytosolic and also partly mitochondrial based. So it has an enzyme that is found on the mitochondria and also found in the cell cytoplasm, right? So that is the cellular compartment. The next outline is the organ of metabolism. Organ of metabolism. In which organs does this pathway occur? This pathway, called gluconeogenesis, occurs in the liver, mainly in the liver, and lesser extent in the renal cortex of the kidney. That is the organ of metabolism. It occurs in the liver and lesser extent in the renal cortex of the kidney. That is the organ of metabolism, right? Okay, we go to the next outline, which is the substrate of gluconeogenesis, or the precursor, right? Precursors of gluconeogenesis. We have four main precursors of gluconeogenesis. The sole aim of these precursors are to produce glucose. The sole aim is to generate what? Glucose. That is the sole aim. The sole aim is to generate what? Glucose. All those precursors are being acted upon by an enzyme to finally produce what? Glucose, which is used for tissue respiration or tissue metabolism, right? So we have the following precursors. One is lactate. The two is glycerol, part of fat. Glycerol, part of fat. Three is gluconogenic amino acid. Genic amino acid. And four is propanyl-CoA of urgent fatty acid. Propanyl-CoA of urgent fatty acid. These are the precursors of gluconeogenesis, whereby an enzyme of gluconeogenesis is acted upon to synthesize glucose for cellular respiration or tissue respiration, right? So that we can generate energy. So these are the precursors of gluconeogenesis. Okay. We have the gluconogenic amino acid. This amino acid undergoes what? Transamination reaction to produce a corresponding carbon skeleton, which is being converted to a certain intermediate in PCA cycle to generate oxalic acetate or pyruvate. Those are the activities of the glucogenic amino acid. Now, what are the examples of the glucogenic amino acid we have in gluconeogenesis? Number one glucogenic amino acid we have alanine, we have aspartic acid or aspartate, and number three, glutamate or glutamic acid. Now, these are the three profound glucogenic amino acid, right? But we have other amino acid that undergoes what? Transamination reaction to form a corresponding carbon skeleton, which is converted into a certain intermediate in TCA cycle to generate oxalic acetate and pyruvate in order to form glucose. Because this is the primary aim of gluconeogenesis, to generate what? Glucose. All these reactions is to form this, which is glucose, right? Okay. Now, among these four precursors of gluconeogenesis, the primary substrate or precursor of gluconeogenesis is the lactate and gluconeogenic amino acid. Lactate and glucogenic amino acid are the primary precursors or the major precursors of gluconeogenesis, whereas glycerol and propanoic-CoA of urgent fatty acid are the minor source of glucose from gluconeogenesis. That means they are the minor precursor of gluconeogenesis, right? Pay attention. Among these three gluconogenic amino acids, only alanine is the major gluconogenic amino acid, right? Because alanine is being channeled to Cahil cycle or glucose-alanine cycle, because alanine is stored in a muscle, right? So during gluconeogenesis, alanine will be mobilized to be transported to the liver. In the liver, alanine will be transaminated to form pyruvate by an enzyme called alanine aminotransferase or alanine transaminase, right? When it forms pyruvate in the liver, pyruvate will be channeled to the glucogenic pathway in order to form glucose. So that is how it works. I will still explain the cycle here called the Cahil cycle or glucose-alanine cycle, right? So these are the precursors. You have to understand that from glucose to pyruvate is glycolysis. Right? Glucose coming down here, because you have in glycolysis, we break down glucose molecule, right, to form energy, right? So when we break it down, from glucose goes downward to form pyruvate is what? Glycolysis, precisely aerobic glycolysis. While from pyruvate to lactate is what? Anaerobic glycolysis, right? That is it. Now, in gluconeogenesis, we are trying to build up glucose. Glucose is being tried to build up, right? That means we have to start from where? TCA cycle, because pyruvate is involved. Pyruvate is the first what? Intermediate that is the first precursor that has to be reduced to form an intermediate in TCA cycle, which is essential for gluconeogenesis. And that intermediate is called what? Oxalic acetate, right? I repeat, glucose, from glucose to pyruvate, like the arrow coming downward is glycolysis. That means this side is glycolysis. Why this side is what? Gluconeogenesis. Because when arrow is going downward, we are trying to break down glucose. That is for glycolysis, right? Now, when gluconeogenesis is occurring during starvation or fasting, or in the case of diabetic mellitus, we are trying to build up glucose back, because in glycolysis, glucose was broken down. But in this pathway, glucose has to build up in order to be broken down again and give us energy. So the step one has to start from TCA cycle, right? Just pay attention. Let's journey together. You get to understand it. It is so simple. Just be attentive. Pay attention, right? Okay. We have discussed the introduction, cellular compartment, organ of metabolism, the precursors, right? Now let's dive into the metabolic pathways before we discuss the significance and the regulations. Pathways. Pay attention, right? The first thing that will happen in this step 1 is that recall that pyruvate is not permeable to the mitochondrial membrane, right? Pyruvate is not what? Permeable to the mitochondrial membrane. It requires a what? A transporter that will transport it from the cytosol to the mitochondria so that it will be reduced or carboxylated to oxalic acetate, and that is the first step of gluconeogenesis, right? Now pay attention. Pyruvate will be transported from the cytosol to the mitochondria by a transporter called pyruvate what? Transporter, right? So this, let's call this pyruvate transporter. Pyruvate what? Transporter, right? Pyruvate transporter. It will transport pyruvate from the cytosol to the mitochondria. So pyruvate has been transported. Once pyruvate has been transported, that is the inception or the statement of the gluconeogenesis, right? Pyruvate will be carboxylated to form oxalic acetate. Carboxylated to form oxalic acetate by an ATP hydrolysis reaction, because ATP is being hydrolyzed. And recall that it is a carboxylation reaction. That means CO2 is added. It is a CO2 fixation reaction. CO2 will be added. CO2 will be added, right? So recall that ATP was utilized. ATP will be reduced to form ADP plus PI, one inorganic phosphate, because ATP is utilized for the forward reaction. That is why it is called an ATP hydrolysis reaction, right? So pyruvate will be carboxylated to form oxalic acetate by an enzyme called pyruvate carboxykinase or pyruvate carboxylase, because CO2 is added. So it is a CO2 fixation reaction. CO2 fixation reaction, right? So the enzyme is called pyruvate carboxylase. Pyruvate carboxylase. Pyruvate carboxylase or pyruvate carboxykinase, whichever one, it is the same enzyme, right? So this is the first step of gluconeogenesis. So it is called the first step of gluconeogenesis. Recall that pyruvate can also be oxidatively decarboxylated to form acetyl-CoA, right? You have to understand that in TCA cycle now, right? But that one is a different reaction. It does not concern gluconeogenesis, although acetyl-CoA is an activator of pyruvate carboxylase. That means when there's an increase in production of acetyl-CoA, there is usually an increase in synthesis of oxalic acetate, because it will also be activating pyruvate carboxylase in order to do what? To carboxylate pyruvate and form oxalic acetate. That is why acetyl-CoA is an activator of pyruvate carboxylase or pyruvate carboxykinase. So we have formed our step 1 of gluconeogenesis, right? But the striking thing is that, pay attention, the striking thing is that oxalic acetate is not permeable to the mitochondrial membrane, because the step 2 of gluconeogenesis occurs in the cell cytosol. So since oxalate acetate is not permeable to the mitochondrial membrane of the cell, it requires a shuttle, and that shuttle is called malate aspartate shuttle or malate shuttle. It is called malate aspartate shuttle, malate shuttle, or malate aspartate shuttle. Aspartate shuttle, right? Now watch. For oxalic acetate to be transported from the mitochondria to the cell cytoplasm so that step 2 will take place, it has to be converted to malate, because malate is permeable to the mitochondrial membrane. That is where this shuttle comes in, called malate aspartate shuttle, right? Now watch. Oxalic acetate will be converted to malate to transport it. And this is catalyzed by an enzyme called malate dehydrogenase. Malate dehydrogenase. That means NADH is generated. Right? So the enzyme is malate dehydrogenase, MDH. Malate dehydrogenase will catalyze the conversion of oxalic acetate to malate, thereby transport it to the what? To the cell cytoplasm, right? Malate will be reduced to form oxalic acetate back, right? This is oxaloacetate, right? By the same enzyme MDH. So this is the shuttle, right? The shuttle, it is called malate shuttle or malate what? Aspartate shuttle. That is when those amino acid comes in, right? So you can see that oxalic acetate has been transported from the mitochondria to the cytoplasm, so that step 2 of gluconeogenesis will take place, because step 1 occurs in the mitochondria. That is why the only enzyme of gluconeogenesis that is a mitochondria based enzyme is pyruvate carboxylase or carboxykinase that catalyzes step 1 of gluconeogenesis. You can see it here, right? So we have generated our oxalic acetate. We have transported it from the mitochondria to the cytoplasm. The next thing that will happen is the formation of step 2, right? Step 2 will be formed. Oxalic acetate will be converted to form phosphoenol pyruvate, right? By an enzyme called phosphoenol pyruvate carboxykinase. And this reaction requires we have GTP will be reduced to GDP plus PI, or GIP or ITP will be reduced to IDP plus PI, whichever way. These are the coenzyme. Important to note, there is a coenzyme that also plays a role in this step 1. It is called biotin. So you can add it there. Biotin is also here. Biotin plays a role of coenzyme in the step 1. So the enzyme that catalyzes this step is called phosphoenol pyruvate carboxykinase. Phosphoenol pyruvate carboxykinase will catalyze the conversion of azotic acetate to phosphoenol pyruvate. That is why phosphoenol pyruvate carboxykinase circumvents the step 9 of glycolysis, right? You can see it here that this is the step 9 of glycolysis, right? This is the step 9 because in this place, ADP will be reduced to ATP by an enzyme called pyruvate kinase, right? That is for glycolysis. Pyruvate what? Kinase. The enzyme for glycolysis, and is the key enzyme. That is why the 4 key enzymes for gluconeogenesis circumvent the 3 key steps of glycolysis, which is catalyzed by its 3 key enzymes. So step 9 is one of those key steps of glycolysis that is being circumvented by the key enzymes of gluconeogenesis. You can see it that phosphoenol pyruvate carboxykinase have circumvented this step 9, right? It has circumvented what? This step 9 of glycolysis. So this is step 1 of gluconeogenesis. So this is what? Step 2, right? Okay. CO2 is also added, right? Now, we have formed our step 2. The next step we'll form is the step 3 and step 4. Now, once this step has been circumvented, there will be a partial reversal from step 2 to step 3, right? There will be a partial reversal of step 2 to step 3 in order to do what? To circumvent this step also. So, recall that in glycolysis, this is one of its key steps, that is step 3, right? So, ATP will be reduced to what? ADP. And the enzyme that catalyzes the step 3 of glycolysis, which is one of its key enzymes, is phosphofructokinase. Phosphofructokinase, right? Phosphofructokinase. But then, in gluconeogenesis, it will be circumvented by an enzyme called fructose 1,6-bisphosphatase. Fructose 1,6-what? Bisphosphatase, whereby water molecule is added and Pi generated, which is what? Inorganic phosphate. The enzyme is called glucose 6-phosphatase enzyme. Fructose 1,6-bisphosphatase. Fructose 1,6. This is the key enzyme of gluconeogenesis, which circumvents the key step of glycolysis, which is step 3, right? So this is the step 3 of gluconeogenesis. This is step 1, step 2, this is step 3. The next thing will happen again is, because our sole aim is to form glucose, we are trying to form glucose, right? Starting from the onset, which is in in the mitochondria in TCA cycle. All our aim is to generate what? Glucose, right? Now, we are trying to form the step 4, which is the final step, and glucose will be finally be what? Be generated in the liver, right? Now watch. ATP will be also be reduced to what? ADP. And the enzyme that catalyzes this step is the isoenzymes of glycolysis, right? Which is either hexokinase or the glucokinase. So let's put hexokinase. Hexokinase. Water will be added. PR will be generated. The enzyme that catalyzes this step so that glucose will finally be generated is glucose 6-phosphatase enzyme. This enzyme is a hepatic enzyme. It is a hepatic enzyme. We call it hepatic glucose 6-phosphatase. It is mainly found in the liver. That is why liver is the only organ that can replenish or influence the blood glucose level, because it is composed of this enzyme. That is to say that in glycogen metabolism, right, where we have the muscle glycogen and the liver glycogen, the reason why muscle glycogen is not used for tissue respiration, that is the energy from it in the glycogen, is that muscle lacks this enzyme. Muscle lacks glucose 6-phosphatase. So those glycogen stored in the muscle is usually used for muscular contraction. But the liver glycogen will be used to generate glucose because liver has this enzyme. That is why this enzyme is the enzyme that tells if glucose will be generated or not, because you can see it here. It's the final enzyme that does what? That act to form glucose. So it is an important vital organ. It is a vital organ. I mean, it is a vital enzyme in this reaction, right? Because it is the enzyme that tells if glucose will be formed or not. And that is why the muscle glycogen is not used to generate glucose. It is only used for muscular contraction because the muscle lacks what? Glucose 6-phosphatase enzyme. It is only found in the liver. That is why it is called hepatic glucose 6-phosphatase, right? So that is how it works. So we have generated our glucose. So this is how the pathways of gluconeogenesis takes place. This remains our TCA cycle. Now pay attention. Let's discuss how these precursors helps in generating glucose. I believe we only listed it, right? Let's discuss it. How it helps, how it participates in glucose production as being the precursor or the substrate of gluconeogenesis. Let's start with lactate. This is lactate, right? Now, as you can see, lactate is found in the muscle, right? So during gluconeogenesis, lactate will be transported from the muscle to the liver. Now, in the liver cell, lactate will be converted to pyruvate by the lactate dehydrogenase enzyme. Pyruvate will be channeled to gluconeogenesis to generate what? Glucose. So that is the fate of lactate being the precursor of gluconeogenesis. From muscle to diffuse to the liver. In the liver, it will be converted to pyruvate by an enzyme called lactate dehydrogenase. Pyruvate will be channeled to gluconogenic pathway to generate glucose. That is how it works, right? And also in glycolysis, lactate plays a huge role in Krebs cycle because the muscle cramp seen in athletes occurs as a result of accumulation of lactate or lactic acid as a clinical condition called lactic acidosis, right? That is in the case of glycolysis. That is anaerobic glycolysis, right? Now, the next one is glycerol. Now what? During glycolysis, glycerol is usually stored in the liver, glycerol part of fat. But during gluconeogenesis, glycerol will be phosphorylated to form glycerol 3-phosphate by an ATP-dependent enzyme called glycerol kinase, right? You can see during gluconeogenesis, glycerol will be what? Mobilized by a phosphorylation reaction to generate what? Glycerol 3-phosphate by an ATP-dependent enzyme called glycerol kinase. So it will form DHAB. From DHAB, it will channel to glycerol, there are 3-phosphate in order to form pyruvate. Pyruvate will follow the pathway to generate what? Glucose. That is how it works, right? That is how the reaction goes. The next one is glucogenic amino acid, where we have the alanine, aspartate, and glutamate. You have to understand that an aspartate here, an aspartate mostly got transaminated, right, to form a corresponding carbon skeleton, which will be converted to oxaloacetate. So aspartate should be here. Aspartate is usually converted to what? Oxaloacetate, right? So that is how it works. For the case of glutamate, glutamate will also be transaminated to form a corresponding carbon skeleton, which will generate what? Alpha-ketoglutarate. Though there are some other amino acids that also undergo what? A transamination reaction to generate acetic carbon skeleton, which will form alpha-ketoglutarate, right? So in this case, we have glutamate, we have histidine, we have proline, we have arginine. These are those amino acids that will generate alpha-ketoglutarate as a result of their transamination reaction during what? Gluconeogenesis. Their sole aim is to generate what? Oxaloacetate. That is their sole aim. The sole aim of this amino acid is to do what? Generate oxaloacetate. And also what? Pyruvate, but mostly is oxaloacetate, so that it will be channeled from this reaction to generate what? Glucose, right? Now, in the case of alanine, I've said previously that alanine is the most potential glucogenic amino acid. It is being stored in the muscle, right? So, during gluconeogenesis, alanine will be transported from the muscle to the liver. In the liver, it will undergo what? Transamination reaction to form pyruvate by an enzyme called alanine aminotransferase or alanine transaminase enzyme, in which alpha-ketoglutarate will be reduced to glutamate. That is their coenzyme, right? So once it forms pyruvate, pyruvate will be channeled to gluconeogenesis, right? To generate what? Glucose. You can see it here. That is why I say that their sole aim is to generate what? Either oxaloacetate or pyruvate, because these are the cardinal components as an intermediate of gluconeogenesis, so that glucose will be generated, right? So that is how it works. But you have to understand that there are differences between glucose-alanine cycle, which is the Cahie cycle, which is the cycle for gluconeogenesis, and also Cure cycle. Cure cycle occurs during strenuous exercise, that is anaerobic glycolysis, right? Whereas Cahie cycle, which is the one that refers to alanine or glucose-alanine cycle, occurs during starvation or fasting, or in the case of diabetic mellitus patient. That is the difference. Because two of these cycles uses the same organ, the muscle and the liver, but of different metabolic profiles. That is how it works. Okay? So the next one we shall be looking at is the propionyl-CoA. Propionyl-CoA, right? You have to understand that This propanil-CoA of all-chain fatty acid is a minor source of glucose, being the precursor of gluconeogenesis, because this propanil-CoA will do what? It will form a certain amino acid, right? Which will now form succinyl-CoA. That is why it mostly linked to succinyl-CoA, right? It is a minor source of glucose, being the precursor of gluconeogenesis. So let's add propanil-CoA here. Propanil-CoA. Propanil-CoA is here, right? So that is how it works. You have to understand that the even chain fatty acid, which is for acetyl-CoA, is not and can never be a precursor of gluconeogenesis. Acetyl-CoA, which is the even chain fatty acid, is not a precursor of gluconeogenesis. It is only odd chain, which is propanil-CoA, because acetyl-CoA is oxidatively decarboxylized in the TCA cycle in step 3 and step 4 as a 2 atom of CO2. That is why it does not follow the forward reaction. You have to understand that the only substrate of gluconeogenesis in TCA cycle is the catalyst called oxalic acetate. Oxalic acetate is a true precursor or substrate of gluconeogenesis in TCA cycle. That is an important clarification, right? You have to note that down. Note it down that this is so essential for this pathway to always take place, right? Because it's an important intermediate that mediates all these reactions, right? So that is how that one works. I believe we have done justice to all these precursors and all that. The next which I'll be looking on to is the regulation. The regulation. Regulation. We have allosteric regulation, allosteric regulation, and hormonal regulation. Now watch. When we talk about allosteric regulation, we refer to those key steps or the key enzymes of gluconeogenesis. You have to understand that the 4 key enzymes of gluconeogenesis includes the pyruvate carboxylase or carboxykinase, the phosphoenolpyruvate carboxykinase, the fructose 1,6-bisphosphatase, and glucose phosphatase. These are the 4 key enzymes of gluconeogenesis which act on the 4 key steps of gluconeogenesis, right? That is an important clarification, and it is one of it allosteric regulation, right? You have to understand that pyruvate carboxylase or carboxykinase is activated by acetyl-CoA. I've said previously that increase in acetyl-CoA pool or increase in synthesis of acetyl-CoA will favor the production of oxalic acetate, because it will always be available to activate pyruvate carboxylase to convert pyruvate to oxalic acetate. That is the metabolic analysis, that acetyl-CoA is the activator of pyruvate carboxylase. That is how it works. Okay. Now, on the case of hormonal regulation, right? There are hormones that counters the activities of gluconeogenesis and also favors the activities of gluconeogenesis. The chief hormones of this pathway include glucagon and glucocorticoid. Now, Glucagon is majorly the chief hormone of gluconeogenesis. It works by activating its key enzymes. So glucagon favors the induction of synthesis of the 4 key enzymes of gluconeogenesis. That is what it does. It favors the induction of synthesis of the gluconogenic key enzymes, which is what we have listed, right? Why the glucocorticoid? What does it do? It initiates the synthesis of hepatic aminotransferases, thereby providing substrate for gluconeogenesis. That is what it does. It favors the synthesis of hepatic aminotransferases, thereby providing the synthesis, thereby providing the substrate for the synthesis of glucose, right? That is what it does. So this hormone is essential, right? It's essential because it provides, it works by activating those hepatic aminotransferases, thereby providing substrate for gluconeogenesis. That is what the hormone does, right? So the next one is insulin is a counter hormone. Insulin is a what? A counter hormone. Insulin is a counter hormone, right? It works by deactivating gluconeogenesis by inhibiting the key enzymes. So insulin is a counter hormone in this reaction, in this pathway, whereas glucagon is a current hormone because this glucagon is the chief hormone of this pathway, whereas insulin is a counter hormone. That means it inhibits what? Gluconeogenesis. You have to understand that glycolysis and what? And gluconeogenesis will not occur at the same time. One must be inhibited, one must be inactive, right? This pathway occurs during what? Starvation or fasting, or in the case of diabetic mellitus, whereas glycolysis occurs during well-fed state. That is their difference, right? So the next we shall be looking on to is the significance. 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 What are the significance of gluconeogenesis? The number one significance of gluconeogenesis is that it has the ability to replenish the blood glucose level. That is the liver, right? Because the liver mostly have this enzyme. That is why it's called hepatic what? Glucose phosphatase. Although we have it in a lesser quantity in kidney and intestinal mucosa, but that is not the case. It is mainly found in the liver. That is why the liver has the ability to replenish the blood glucose level by influencing the blood sugar level. So the significance of this pathway is that the liver is the only organ that replenishes the blood glucose level by influencing it, because it has this what? Enzyme called glucose phosphatase enzyme, which can be called hepatic glucose phosphatase enzyme. So that is how it works, right? Now, that is significance. Watch here. There are some other amino acids that also got what? Transaminated here, such as methionine and also valine. So these are those amino acids, right, that got also transaminated just to form a certain carbon molecule. It's a certain carbon skeleton that will be converted to succinic OA. They are so aiming to generate oxaloacetate or pyruvate. So we have come to the end of this class. You have to understand that this is the step 1 of glycolysis, right? This is step 3. We're noting those key steps. Why here is step 9. So we can see it. 3. Here remains step 4 of gluconeogenesis. So this is step 1 of gluconeogenesis. Step 2 step 3, and step 4. Whereas this is step 1, step 2, and step 9 of what? Of glycolysis per se, please. Right? So that is how it works. So you can see that the enzyme of step 4 circumvent the key step of glycolysis, which is being counted by the step 1 key enzyme of glycolysis. You can see it there, right? That is how it works. I believe we have done justice to gluconeogenesis, starting from the introduction to the significance and also the regulation, right? Thank you so much for staying to the end of this class. I really appreciate it.